Hello, students. This is a lesson, this is a recorded lesson, which is specially recorded for those who were absent yesterday, Saturday. So basically, yesterday we actually learned on objective number 9. Okay, objective number 9, which states describe the structure of cholinergic synapse and explain how it functions, including the role of calcium ions. Okay, the role of calcium ions. So we're going to look at the structure of cholinergic synapse, what type of synapse, cholinergic synapse, and explain how it functions, including the role of calcium ion. Okay, so let's look at okay, how does the nerve impulse pass from one neuron to the next neuron. This one we talked about already last Thursday, where the electrical impulse may propagate, along all of the sections of the axon. That is for the non-myelinated, but for myelinated, it will just simply jump, from one node to the next node. Okay, and that is for the myelinated. So you can revise on that content regarding that learning objectives last Thursday's lesson. Okay, so today's learning objectives about the synapse, and the syllabus states that you only need to know cholinergic synapse. Okay, so there are many. First of all, there are many different types of synapses. There's synapses that inhibit. There are also synapses that stimulate, or enhances. For example, if you did the MCQ, synapses or neurons that release GABA, and the neurons that release serotonin, and there are also neurons that release acetylcholine. And so those neurons that release acetylcholine are known as cholinergic synapse. Okay, cholinergic synapse, because they secrete acetylcholine. Is either the nerve secretes acetylcholine, or the nerve is stimulated because of acetylcholine. Okay, so when we talk about synapse, we're actually looking at communication. What happens to the action potential when it arrives at the synaptic node or synaptic terminal, and how does, if you look at this particular image, what happens to the nerve impulse when it arrives at this terminal, synaptic node or synaptic bulb, and how does action potential occur in the next neuron? Okay, the next neuron. So, and this is what we're going to learn about synaptic transmission. Okay, so you need to know about some terms. For example, pre-synaptic neuron, and then post-synaptic neuron. So pre and post, So basically, pre means, the word pre means before or previous, whereas the word post means after, or basically the next one. The next one. Okay, so when you look at this particular slide, I can just clear this. This is the pre-synaptic neuron, the previous neuron, and this is the post-synaptic neuron, the next neuron. Okay, so this can be between two motor neurons, how two motor neurons communicate, or how two sensory neurons communicate, or between sensory and relay neuron, or between relay neuron and motor neuron. All right. So our main purpose today, today's lesson is communication. How the arrival of nerve impulse at the synaptic knob causes action potential and the continuation of the nerve impulse in the next neuron. Okay. So we say just now that this is so if we zoom, we can now zoom. Okay, we can now this is synaptic, this is now we can now be more specific, pre-synaptic membrane. Okay, and then the dendrites, this is the post-synaptic membrane. Okay, the membrane of the previous neuron, and then it's the membrane of the next neuron. So presynaptic membrane, postsynaptic membrane. Okay, so this is of what? Of synaptic bulb, or sometimes known as knob. Okay, knob. And whereas this one here is postsynaptic membrane of the dendrites, yeah, of the dendrites. And then of course the cell body, yeah, the cell membrane, the dendrites, because the dendrites actually continuation, yeah, with the cell membrane of this cell. This is cell body, also known as soma. Okay. All right. So here is another image. Okay. So basically, there is a gap here, yeah? A gap between the presynaptic membrane. Yeah. This is the synaptic knob, presynaptic membrane, and the cell membrane of the postsynaptic membrane. There's a gap here. So this gap is a small gap here. It's called synaptic cleft or synaptic gap. Yeah, synaptic cleft or synaptic gap. Okay, and when you look at this particular slide, you can see that we can already see the role of synapses. Yeah, there are many roles of synapses, but from this particular slide, you can already tell one role of synapse, and that is it acts as junction. Okay, so this particular neuron may receive incoming impulse from other neurons, so it serves as a junction, okay, to relay the nerve impulse to other neurons. Okay, so the meaning of pre and post. Pre means before. Okay, or previous. Okay, the previous one. Post means after or the next one. Yeah, next one. That's the meaning of pre and post. Okay, so now this is the enlarged. So what you see, this one here, is actually this one here. Yeah. So what you see there. So let me just enlarge this particular area here. Yeah, or it can also be this area here, or this area here, or this area here. Yeah. Okay. So, and of course, this area here, this area here. Okay. And that is what you see here. Okay. So this can be this is the presynaptic membrane of the synaptic bulb or the synaptic knob, and this is the postsynaptic membrane of it can be the dendrite, it can be the cell membrane of the neuron. Okay. So when you look at the terms, see presynaptic neuron, postsynaptic neuron, and this is the membrane, presynaptic membrane, postsynaptic membrane. Okay. The synaptic cleft or gap. Okay. Synaptic bulb or knob, depending on which textbook you are referring to, or terminal. And now you see the vesicles, which contains this vesicle here, which contains acetylcholine. Okay. And of course, lots of mitochondria. Okay. And then this is the receptor for the acetylcholine. Okay, receptor for the acetylcholine. Now, you see there are lots of mitochondria here. So immediately a lot of students think that, this may be associated with active transport. Yeah, but this is not the case. Okay. And you will learn later on that many mitochondria here in the synaptic knob is not for active transport. It's for something else, and we will look at that later on. Okay, these are just taken from different textbook. You can just have a look here. Even this one as well. You can see it's the same thing. Okay, synaptic vesicles, which contains acetylcholine. Yeah, this is acetylcholine, the neurotransmitter acetylcholine. So it's called cholinergic synapse because there are other different types of neurotransmitters that release serotonin, that release GABA, and these have been given different names. It may not be cholinergic synapses. Okay. Right, so this is also taken from a different textbook. Right, now I just want to highlight this area here. Yeah, so you can see this vesicle will fuse. All of this vesicle here will fuse with the presynaptic membrane. So the term fuse is very important, and then the term exocytosis is also very important. Yeah. So when the vesicle, which contains acetylcholine, fuse with this presynaptic membrane, the acetylcholine is released by exocytosis into the synaptic cleft or synaptic gap. Okay, and then it will bind to its receptor. So this is the receptor here. Receptor, yeah. So this part here is the receptor. This receptor of acetylcholine. And when the receptor binds, when the acetylcholine binds to the receptor, so you can now see that what happens next is sodium channels are open. So when sodium channels are open, the sodium will enter. So this is the next event. And so when the sodium enters, depolarization will occur in the next segment here, which is in the postsynaptic neuron, yeah, whereby the inside will become positive, okay, compared to outside. Yeah, inside will become positive compared to outside. So action potential will occur. Nerve impulse will occur in the next in the post-synaptic neuron. Okay, so it's just some of the images. You can spend more time when you look at the actual soft copy. Yeah, as you can see here, these are the vesicles containing acetylcholine. This is the gap, synaptic cleft. Okay. All right. So now this is the most important one. We're going to look at the events, meaning the sequence of events, step 1, step 2, step 3, and so on. What occurs at the synaptic bulb or synaptic knob. So first, now that you already know the structure, okay, so before that, there is an additional structure here which we have just mentioned just now, and that is the sodium channel. Okay, this is the receptor for the acetylcholine. This is the sodium channel. Okay, so don't forget about that as well. Okay, so what happens is that this is the arrival of electrical impulse or nerve impulse. So when there is an incoming electrical impulse here, what happens is that before this vesicle fuse with the presynaptic membrane, yeah, the arrival of this nerve impulse at the synaptic knob will cause calcium to enter. Yeah, will cause calcium to enter. This is very, very important because you have it stated, yeah, in the learning objective, including the role of calcium. Describe the structure of a cholinergic synapse and explain how it functions, including the role of calcium ions. Okay, so you need to know the function of calcium ions. So what is the role of calcium ion here? The role of calcium ions, so we're going to study it in a moment. So the arrival of nerve impulse, okay, the arrival of nerve impulse at this synaptic knob will cause calcium ions, first of all, calcium channel open, and then will allow the entry of calcium. Okay, and this is already one role. Now that you know another role of calcium apart from the strengthening of bones, but calcium is also involved in nerve transmission, and that is at the synapse. Yeah, at the synapse. So it's not only for muscle contraction, it's not only to strengthening your enamel, your bones, yeah, but also in synaptic transmission. The same thing when we look at sodium. Yeah, sodium. Sodium is not only to regulate osmotic balance of water in our blood, but sodium is also required in synaptic transmission. Okay, so that's another role for sodium. Potassium as well, calcium, sodium, potassium, as well as chloride ions. Okay, so the arrival of nerve impulse will cause the calcium channels to open, and then calcium will enter, and that will cause the vesicle to fuse with the presynaptic membrane. Okay, so this vesicle containing acetylcholine will fuse with the presynaptic membrane, and it will release the acetylcholine, and it will bind to the receptor, which is here. Okay, so the binding of this acetylcholine to its receptor will cause these sodium channels to open, and so sodium will enter. So yeah, that's the next event. And now you can see that the inside will be positive as compared to outside. In other words, action potential occurs. And then of course, then local electrical circuit will occur, whereby positive zone will be attracted to negative zone. Yeah, or if it's non-myelinated, then it will go through all the sections. If it is myelinated, then it will jump. Okay, saltatory conduction. Okay, so you can see already what I've mentioned. All of the wordings are here. Don't forget the term fuse, the term exocytosis. Okay. Right. So the binding of acetylcholine to its receptor will cause this sodium channels to open, sodium will enter, and so as you can see here, there is an impulse already. It's actually positive now here is already positive, outside negative. Okay, in other words, action potential occurs here, and then it will propagate along the axon. So if it's non-myelinated, it will have to pass through every section. Yeah, every section. If it is myelinated, then it will jump. Yeah, it will jump saltatory conduction. Okay, now that is not the end. Yeah, so what happens after action potential occurs? So action potential already occurs, and the action potential will be gone. Yeah, and then it will be ready for the next action potential. So to prepare for the next action potential, this acetylcholine must be recycled. It cannot continue to be bound to its receptor. This acetylcholine must be broken down because it needs to be recycled. It needs to go back to its presynaptic membrane so that it will be ready for the next action potential. Okay, so there is the next one is to break down the acetylcholine by this enzyme cholinesterase. Now, this enzyme cholinesterase are found on the postsynaptic membrane. Yeah, these enzymes are found on the postsynaptic membrane. Yeah, and this enzyme will break down acetylcholine to acetyl and choline. Okay, so it needs to be broken down so that it's made easier, smaller. It's broken down into acetyl and choline so that it's easier for these substances to diffuse back. It is made smaller and perhaps maybe lighter, yeah, so that it will be easier for it to diffuse back to the presynaptic membrane or neurons. Okay, so basically we have acetylcholine in the presence of the cholinesterase will be broken down to acetyl and choline. Yeah. Now this acetyl has many names. Yeah. It's also called acetate. It's also called ethanoic acid. Okay, acetyl or acetate or ethanoic acid. Yeah, so these two will diffuse back to the presynaptic neuron. Yeah, it will diffuse back. So why is it broken down? It's because it's easier. It will be perhaps lighter, so it will be faster diffusion. It can diffuse faster to the presynaptic membrane or neuron. Okay. Now once the acetyl and choline, okay, so now I write it down here. Once the acetyl and choline is in this area here, to join these two together requires ATP. And this is the reason why there are lots of mitochondria. So the mitochondria is not for active transport. Okay, the mitochondria is not for active transport, but rather the combination of acetyl and choline to form acetylcholine requires ATP. Okay, and that's it says here, this requires energy, ATP. So the reformation of acetylcholine requires ATP, which is provided by the mitochondria. Okay, so that's it, yeah, for the events. It's very simple actually, and it's very the events are very sequential, in a very sequential manner. A lot of students they just need to, do not skip the steps. For example, fuse, exocytosis, synaptic cleft. Okay, these are very, very important binds the receptor, and then sodium channels open, and of course don't forget about the calcium as well. Yeah, the calcium ions. So everything must be in sequence. You cannot miss the important points. Okay, and don't forget about the recycle of the acetylcholine. Don't also forget the name of this enzyme, cholinesterase. Spelling is also very important. Cholinesterase. Okay, this is taken from a different textbook, so it's up to you. You want to look at it. So it's up to you. Yeah, I'm not gonna repeat it again. So just read it in your own time. This is again taken from a different textbook, so you can see there are other names of the acetal. It's also known as acetate. Okay, also known as acetate. In this textbook, it's also known as ethanoic acid. Ethanoic acid. So there are many names. Acetal. The latest one, of course, according to textbooks, they call it acetal. Don't forget, it's also called acetate, or ethanoic acid. When it's broken down, when the acetylcholine is broken down to choline and acetal or acetate or ethanoic acid. Okay. Right. Here, what I want to highlight is this one here. This is the thing about having different resources. So you can see where the enzymes can be found. The enzyme acetylcholinesterase is attached to the postsynaptic membrane. Okay, attached to the postsynaptic membrane. So the enzymes are found on this postsynaptic membrane. Yeah, postsynaptic membrane. Because this is where the acetylcholine will bind to its receptor. Okay, these are just some notes that you can read in your own time. To understand to you about the different types of neurotransmitters. You can read in your own time. See the GABA, and then the dopamine or serotonin. Yeah, there are many different types of neurotransmitters. The MCQ as well. Okay, so on Monday we're going to look at neuromuscular junction. Okay, so what we have studied here is communications between neurons. So it can be between two sensory neuron, or between two motor neuron, or between sensory and relay neuron, or between relay and motor neuron. How action potential is resumed, when it reaches the end of a neuron here. How does the action potential or nerve impulse can continue in the next neuron? And that is about synaptic transmission. Now, if the nerve impulse, eventually the nerve impulse will reach an effector. So when it reaches this nerve impulse, eventually will reach an effector. An effector, it can be muscle or gland. Then this is called the neuromuscular junction. Neuromuscular junction, and this is something that we're going to cover on Monday. Okay, neuromuscular junction, meaning now it's the communication between the presynaptic membrane of this particular motor neuron and the cell membrane of the muscle, which is called sarcolemma. Okay, sarcolemma. And what are the communication here? Is it the same as synaptic transmission, or is it different? And that is something that you will learn on Monday. So this is the neuromuscular junction, because eventually the nerve impulse will reach an effector or a gland. So how does communication, how do they communicate? What happens here in this part here? And that is something that we're going to look on Monday. Okay. Very interesting images here. As you can see, the neuromuscular junction. You can see nerve here will terminate at the muscle. sarcolemma, the membrane of the muscle. This one as well, very, very interesting image. We'll look at this image again on Monday. All right, now what is the function of synapse? Yeah, now this is very interesting. Apart from junction, I did mention that it serves as junction, and this particular nodes, you can see here, act as junction. Apart from junction, it also ensures unidirectionality. Yeah, unidirectionality means it ensures that nerve impulse will only travel in one particular direction. So let's look at this one here. Okay, so nerve impulse will only travel in this direction, yeah? In one direction. It cannot go in reverse. Okay, why it only goes in one direction? Because the acetylcholine is produced in the presynaptic membrane, and the receptor for the acetylcholine is only found in the postsynaptic membrane. Yeah, so the acetylcholine is produced in the presynaptic membrane or presynaptic neuron, and the receptors are found on the postsynaptic membrane of the postsynaptic neuron, and that ensures one direction. Yeah, it will only propagate in one direction, unidirectionality. Okay, and then that's one purpose of neurons. The other one is fatigue. Yeah, fatigue. Now, how do we explain this fatigue? Let me show you, or to prevent overstimulation. Okay. Right. So we have a neuron here. I'm just going to draw very simple. We have one presynaptic neuron and postsynaptic neuron. Yeah, this is the presynaptic neuron. This is the postsynaptic neuron. Okay. So because there is a gap here, there is a gap. Yeah, synaptic cleft. Synaptic cleft, and because of the reactions that occur here, yeah. So, for example, the arrival of nerve impulse in this region here, will cause reactions in this synaptic cleft. That means this neuron can rest. Yeah, because remember, during muscle contraction, there is the action potential and nerve impulse is transmitting or propagating along this region here. Okay, and so if there is a gap here, that means that this neuron can rest for a while, because it will take for the acetylcholine to bind to the receptor. Even though it's, of course, in reality it will be very, very fast, but milliseconds to these neurons are very, very important. Okay, so incoming action potential here, that means this neuron can rest because of the gap here. Or vice versa, okay, meaning when action potential occurs here, okay, action potential occurs here, now this can rest, yeah, can rest. Why? Because it will take time for the acetylcholine to be recycled back. Yeah, acetylcholine needs to be recycled. Okay. So imagine if it is a very long neuron, yeah, where there is no synapse, then that means there will be overstimulation of action potential or nerve impulse, and this can be very, very fatigue. It can cause fatigue, yeah. Because when you have continuous firing of action potential, it will consume a lot of energy. A lot of the sodium potassium pump is doing work to restore the resting potential and so on. Yeah. So at least the presence of the synapse will allow it to rest for a very brief moment, and that is the meaning of fatigue, yeah, to prevent overstimulation or fatigue. Okay. The next one, the filtering out of low-level stimuli. Okay, this is another interesting one. So remember when in a let's draw again another presynaptic neuron and post-synaptic neuron. For action potential to occur, it must reach a certain threshold value, and the threshold value is about minus 55 millivolts. About minus 55 millivolts. Here the action potential, the threshold, meaning the stimulus must have a minimum strength in order for action potential to occur. And this is the reason why you are not aware if small creatures like ants crawl on your skin, yeah, because they are very, very small. You know, the weight, yeah, is not strong enough to cause action potential to occur. Yeah, it's not strong enough to be sensed by the receptor. Yeah, so the stimulus must be around at least minus 55 millivolt or higher. So below this, it will not cause action potential. Now let's say a stimulus occurs just enough to reach the threshold value. Yeah, just enough to reach threshold value. So action potential will occur here. A nerve impulse will propagate here. But the minus, because of the the stimulus is just threshold, it has just reached threshold value. That means maybe small amount of acetylcholine will be released here. Only small amount of acetylcholine will be released, and so not enough to cause action potential in the next neuron. Okay, so that is the purpose of the synapse here. The synapse here also it will filter. It will only allow strong stimuli. It will only allow action potential to occur coming from strong stimuli, not low-level stimuli or weak stimuli. Yeah, and this is very, very important, especially for us human beings. Yeah, we don't want to become oversensitive. Oversensitive means you don't want to, for example, you don't want to react to even very low or weak stimuli, like sound, for example. You don't want to be able to hear low or weak sound. Yes. For example, people, it says here a good example from here. Let's say somebody is doing drilling work. Yeah, say traffic, background noise of traffic 1 mile away, or doing drilling work, yeah, which is 1 mile away. You don't want to be able to respond or hear to this noise, yeah, to this noise. And so that is good. The synapse, the presence of the synapse will prevent that. And that means you will only be responding to important stimuli or high intense stimuli, high enough medium-sized stimuli, at least, yeah, the minimum. You will only be able to respond to at least medium-sized stimuli, to, of course, high level of stimuli. Okay, so that's another role of that synapse. And the next one is learning and memory, and this is very, very interesting. Learning and memory. Learning in memory here. For example, if you are, I already explained this to the previous student. Let's say you are trying to memorize a certain information, and this is your brain. Okay? So let's say, of course, in order to retrieve this information, it requires many different connections, yeah, of different neurons, of many neurons, yeah, of course, sensory neuron, motor neuron, yeah. So and then let's say you're trying to memorize this information, and this is the way you memorize this information, meaning it took you some time to recall, yeah. You might be thinking, oh, it was around, maybe about a few days ago. Was it Tuesday? Was it Wednesday? Was it morning, afternoon? Oh, yeah, yeah. And then you then realize, yes, I was confident that it was on Tuesday, something like that. Yeah. And then you manage to reinforce that information. And what your brain does is that there are neurons here. There are neurons present here. Okay, neurons. And what it does that is that it will build new connection. It will build new synapse. So instead of going zigzag like this, then it will just be in a straight line, because you are able to remember it very, very quickly. Why are you able to memorize it very quickly now? Because your brain has formed new synapse between the neurons, between the existing neurons, and that's why you're able to recall that information faster. So that's about learning and memory. They are, for example, create new pathways and networks. This is like a very good example if when you do sports. When you do sports activity, like one stimulus will cause many different actual potential to occur that will lead to different effectors. Yeah, for example, this may be your muscles, your legs, okay, your muscle, your legs, and then decision making, meaning your brain, inside your brain. Yeah. So for example, if you receive a shuttle, like I myself, I play badminton. If you were to receive a shuttlecock from a server, yeah, what are you going to do next with that shuttle? Yeah. So when you receive the shuttle, you will need to flex your arm, you will need to move your leg, you will need to make a decision as to where you're going to put the shuttle. Yeah. So it's like that. Okay. And the same thing with this one here. This is like, again, it's like, for example, if you're trying to make decision making is always in the brain. Okay. So in order to make decision, you need to receive stimuli from different senses. For example, you need to be able to see, you need to be able to listen. Yeah. You need to be able to feel, yeah. See, listen, feel, all in one, and then that will cause you to make an important decision making. Okay, so it's like that. So this is not in the syllabus, but I just feel that it's good to know as well, because it's very, very interesting. When you look at the syllabus here, it doesn't state anything about functions of explain how it functions. How it functions mean the events of the synaptic transmission. But what are the uses of the synapse? I think that's also important. Yeah, like unidirectionality to prevent fatigue, yeah, learning and memory, act as a junction, and so on. These are important roles of or functions of synapse. Summation, this is no longer in the syllabus. I'm not going to waste our time to spatial and temporal, no longer in the syllabus. So don't bother reading it. Yeah? If you want to know, just read it, just to increase your knowledge. Okay, so that's all for today. So wait again for the next recorded lesson for Monday, yeah? And that will be on the neuromuscular junction. Okay, thank you very much.